What if that next vacation you take has the ability to change your DNA? What if a change in the environment around you, better yet, what if a change in lighting could actually shape the way genes are expressed in your body? While humans might not be able to do that, we recently discovered that some fish are capable of that type of change. And why does it matter? Well, while it might be changing in a fish, it doesn't mean that those same mechanisms and processes don't exist in you and might in fact be perceiving your environment and making very similar changes. My name is Sebastian Alvarado. I'm faculty in the Department of Biology at Queens College with the City University of New York.
Hello, I'm Dr. Wei Fang, a faculty member at Pace University. I'm really excited to talk to you about a study we just published as a research associate at Queens College. This work was made possible by the support of the National Science Foundation to Queens College and Pace University.
Our group, the Alvarado Lab at Queens College, is primarily interested in how we change. We're interested in how the environment has the ability to change your biology.
Our research group is very interested in how animals change the way they look and act depending on their surroundings, a flexibility known as phenotypic plasticity.
If I wanted to study these things perfectly and understanding them perfectly in humans, the best way to do that would to enroll several hundred pairs of identical twins, ask them to participate in studies, and then of course donate all their organs at the end of those studies. So we have to work with the next best thing, which is probably a vertebrate model system. And in that category, we find ourselves turning to African cichlids from the East African Great Rift Lakes.
Within about 10 million years, we saw 2,000 different species arise, which is really quick on evolutionary timescales.
And we work with this one species because it's capable of doing all kinds of really cool things related to its social behavior as well as its colors.
Although we know a lot about the genomes, that still cannot explain the level of diversity. So we wonder, what other processes might be possible to explain the level of diversity, not only the diversity, but also plasticity and how animals can change their colors, their appearance, and the behaviors. Now, within the family of 2,000 species, our study focuses on one species which has the really incredible ability to change colors. They come naturally in between blue or yellow, especially among males.
All you need to know about this fish is that it is capable of really incredible change. It changes with its social environment, it changes colors, and we specifically study males because they have these really beautiful blue and yellow body types.
We found their colors can be controlled and changed in a lab setting when they are raised on different gravel substrates. In a really simple experiment, we first created a blue visual environment on blue gravels for about a month, then move it to yellow gravel substrate for another month, and then move it back to the blue ground for additional month. We were able to control pretty much 100% of their color. Each of these rows represent the same fish that underwent this paradigm where all we changed was the visual environment. You can see this fish went from dark blue to this bright, conspicuous yellow and back to blue again.
So these blue and yellow morphs are really interesting to us because the same male can be one color or the other. And we started to control this within our study by just changing the color of the gravel. Think about it that way. You're living in your home and all I do is I change a bit of the furniture and it has the ability to turn on and turn off genes in your body that reflect how you adapt to that environment. It's pretty mind-blowing if you think about it. And what we found is specifically within this fish, when we changed the gravel substrate from blue to yellow, the fish would, of course, change from blue to yellow. And not only did this happen in the way that you would see it on the outside, but you were also able to see that the pigment cells that allow it to be yellow or blue were changing in their density, but they were also changing in their way that they spread pigments and the way that they show their colors.
To ask how exactly this happens, let's zoom in to the actual substrate responsible for the change.
We decided to resolve that change to a specific molecular mechanism. The molecular mechanisms that we study in our lab fall under the broad umbrella as epigenetics. What does that mean exactly? Well, there's many different things you can focus on. We focus on this process called DNA methylation, which is essentially a molecular off switch. Whenever you see more of it around a gene, it typically turns that gene off. And whenever you see less of it around a gene, it turns that gene on. So we basically study how our environment shapes DNA methylation at many different genes and how that can then turn on and turn off certain traits, specifically within cichlid fish. What causes a fish to change color? In addition to some of these molecular processes, we also look at the cells that are responsible for pigmentation. Our yellow fish are yellow because they have many yellow pigment cells. But not only that, they also disperse pigments in those yellow pigment cells at a far greater degree than blue fish that also have yellow pigment cells. But those blue fish, they hide that pigment because these pigment cells exist either between a dispersed state or an aggregated state.
If we manipulate specific genes and their products that are responsible for hiding or showing their pigment, we would be able to identify the gene candidates that regulates coloration of this animal and what turns these genes on and off. Within this scope, we identified several candidate genes, specifically the endothelin B receptor, and we found that drug pharmacology that targets this receptor only affects the yellow pigment cell.
In our blue fish, we measured much fewer yellow pigment cells. In our yellow fish, we measured many more, but it wasn't just the number of pigment cells, it was their ability to show that pigment, to show it and disperse those yellow pigment granules in those yellow pigment cells. So what exactly is happening inside of these yellow pigment cells that is controlling how much yellowness gets shown in these specific cells? We specifically studied DNA methylation of a gene that's responsible for signaling how pigments get spread out and kind of contracted within those cells. And what we found out is that that specific gene was getting painted with DNA methylation to turn it off when it was yellow. On the other hand, when those fish were blue, it was removing that DNA methylation, removing that off switch, and in turn turning it on. And what we figured out is that it was a way for these yellow pigment cells to become extra sensitive or insensitive to any amount of trigger that causes that aggregation or that dispersal of pigment within those cells. So what does this mean? It means that our yellow fish can become blue, our blue fish can become yellow, and part of it is due to this gene being turned on and turned off. And what we basically think is that this is a starting point to understand how our environment, but not just our environment, but our visual environment can shape our own biology.
Think about it. This is really incredible. We tend to think of animal traits like color as fixed. You're born with it, it's in your genes, and that's that. But this study shows it's not that simple. It reviews a striking example of epigenetic control where the environment dynamically shapes traits we assumed were hardwired. It's as if the environment is talking to the fish's genes. That's an astonishing example of how what an animal sees can influence how the genes were read and expressed.
Now, this is something we probably wouldn't be able to do with humans, and it's really important for us to find all kinds of animals that are capable of incredible changes so that we can start to learn how we change as well, because we do. At one point in your life, you were a single cell embryo. Now you're probably some 37.2 trillion cells. How did you go from a single cell to 37.2 trillion cells with the exact same genetic starting point? Because guess what? Every single cell in your body now shares a genome with that single cell embryo, but you've changed in incredible ways. So how did that happen? And that's really where this whole field of epigenomics helps us measure how our environment shapes our biology. And while it might make sense for how a fish changes from blue to yellow, it's also important to note that it also affects you when you're eating healthy or eating unhealthily, when you're taking part in bad habits that are going to affect your health negatively or positively. It's really, really important to understand how that environment is shaping your biology. This fish, all it had changed was the color of gravel that it swam on. It still ate the same things. It still swam as much as it did before. But this alone, the change in the gravel color, was capable of changing the way they look completely on the outside, to the point where some people might mistake them for two different species, even though it's the same one. It really makes you wonder what in your environment might in fact be shaping your biology.
And if that's true for fish, what else is out there? What other hidden switches are waiting to be flipped in nature?
Thanks for watching this video. If you want to find out more about our work, please visit our lab website, alvaradolab.com. Thanks.